Um, can you see my full screen?
Yes, I can see your screen. Thank you.
It's a full one, right?
Yeah, we can see it.
Okay, good.
Thank you. Thank you.
No worries.
And thank you for accepting to give a presentation late in Australia. I know it's
Well, look,
thank you so much.
it's actually early in the morning because it's
Oh,
it's 2 minutes for midnight. So
okay. So almost.
yeah, it's quite late. It's quite late. I'm happy to do it, you know.
Thank you.
But I was thinking, you know, half an hour ago, I was thinking what I was thinking when I said yes. Because it was 11:30 p.m. So yeah.
We appreciate it. We appreciate it, Professor Carlos. That's so nice of you.
Yeah, no. That is fine. And I can see a couple of members of my lab as well. And probably they're gonna hate me tomorrow because I made them to join the presentation in the middle of the night.
Yeah, that's, you know, that's the problem we have, like with some countries like yours in Australia and other places to accommodate everybody.
Exactly.
It's not easy, but we really appreciate that. Thank you so much. And we're
I'm
gonna
more happy to wait for a couple of minutes before. I know how many people you're expecting, but I'm more than happy to wait.
Yeah, we just give it more minutes, and also to allow Alex to join us, because Alex is going to be moderating the session. We'll be introducing you and moderating the Q&A session.
Okay, great. Thank you.
You're welcome. Yeah, I can see that more participants are joining now. Yeah, more participants are joining, Carlos, so we'll give it more minutes.
That's fine. Yeah.
Hi, Carlos.
To wait. Hey, Alex, how are you?
Alex, good afternoon.
Can you hear me?
Yes. Hi, Carlos. Can you hear me?
Yeah, I can hear you. Yeah. Can you hear me? Yeah.
Yes.
Okay,
Welcome
cool.
to the session. I'm well, thank you. And you?
Good. I don't know if we planned this like this, but you know, here is essentially midnight. It's 12.
Oh my goodness, I'm really sorry. I think that we were trying earlier.
No. That is totally fine. I will just mention that to Latifa
Yeah.
Half an hour ago, I was thinking, actually, what I was thinking when I said yes.
No, we really appreciate it. We know that you're in Australia, and we asked you and you said yes. We said okay. It's especially that we missed last November. We missed it.
Exactly.
So we said, but again, we really appreciate that. Okay, I think
That's all right.
we don't wanna leave people who are joining, and it's really late evening from their end. So maybe we can start the presentation. Alex, what do you think? You can start introducing Carlos if possible.
Okay. Great. So good morning, good afternoon, good evening, close to midnight to those of you or just about midnight for Carlos. Welcome to this webinar. It's a real pleasure to introduce Professor Carlos Salomon, who is a collaborator of mine, and we've known each other for a few years now. Carlos is the head of Translational Extracellular Vesicles in Obstetrics and Gynae-Oncology Group and NHMRC Investigator Fellow, and is considered a worldwide authority on extracellular vesicles as biomarkers for complications of pregnancy, and in ovarian cancer, which is how we had met. He has over 165 publications and a lot of citations of the work that he has done. In the last 10 years, Professor Salomon's primary research and commercialization activities have focused on identification and validation of biomarkers and development on an in vitro multivariate index assays for clinically relevant complications, such as ovarian cancer, obstetric outcomes, obstetrics syndromes, and translating them into clinical application, which I guess is what we are all here to do. He's a pioneer on investigation the release of extracellular vesicles by the placenta and tumor cells and their utility as a biomarker for a wide range of complications that we've just mentioned. So today, Carlos will be talking to you about the burgeoning field of extracellular vesicle signaling and how it holds potential to revolutionize our understanding of cancer biology and really offers non-invasive opportunities and also therapeutic intervention. So I'm probably going to stop here and hand over to Carlos to talking to us about cancer progression, metastasis, response to therapy, and how in early detection EVs can impact on cancer, but also talking about his, I guess, case for extracellular vesicles, translating science from bench to bedside. So over to you, Carlos, and thank you so much for joining us very, very late.
Thank you, Alex. Thank you for the opportunity to share a little bit what we have been doing essentially in our program for the last 10 years. I really appreciate the invitation and apologize in advance because I think we advertised this presentation in November last year, but we couldn't make it. So, it's better, now that, never, right? So I appreciate the invitation and I'm more than happy to give the presentation. So also thank you, Alex, for this very, very, very nice introduction. I really appreciate, the words and highlight essentially what we have been doing in terms of the extracellular vesicle research for several years now. So I'm going to start with something very simple, just to introduce a little bit where we are based as a group. So as Alex mentioned, I'm based at the University of Queensland in Brisbane, Australia. And I have been there for the last 10 years. So we are based in the UQ. This is from UQ is University of Queensland Centre for Clinical Research. And our lab, our program actually function there in the level 7. And this is a very special type of facilities and laboratory because one of the reasons that actually I moved from Chile to Australia 10 years ago was to establish a quality management system in the building. And that quality management system have been actually one of the key aspects for our progression, for the progression of the program. And a key aspect to engage not only with government funding, but industry and commercialization as well. So we're complying with the ISO 17025 for research and development, and we have an entity in Australia, which is the National Association of Testing Authority, what is an independent entity that actually certify that we have the expertise and capability to essentially perform the experiment that are part of the scope of our accreditation. And obviously, one of the key aspects of our accreditation is the isolation characterization of extracellular vesicles, proteomic analysis. So we're trying to identify where are carrier for this extracellular vesicle, including proteins and nucleic acids, and different other methods that are part of the scope of the accreditation. So this is essentially where we are based, and we spend a lot of our time there in the lab. So we are also quite special because we are part of the Faculty of Medicine of the University of Queensland, and we are connected. We have actually a bridge that connects the center with the largest hospital in the state, the Royal Brisbane and Women's Hospital. So we are connecting directly with patients, and there are plenty of different academic clinicians working in the UQCCR, and we have very good interaction with them as well. So this particular center is set up essentially for clinical research, and hopefully to decrease the gap between bench discovery and clinical translation. So in that context, we focus in extracellular vesicle, and here on your right, you can see a general description of our program. I'm gonna timer this because I guess I have probably around 40 minutes for the presentation. You can see people working in extracellular vesicle have a similar kind of structure. So we have been interested in analyze and isolate extracellular vesicles from different biological fluid, including plasma, serum, ascites, saliva, and also from cells. Usually we isolate EVs from cell conditioned media. And what we do is isolate the EVs and characterize them. So I'm gonna go through the presentation to define what is our understanding about the definition of extracellular vesicle, trying to point out also that there are different types of EVs, so define the diversity of extracellular vesicle also is important for clinical translation as well. And as I mentioned, the extracellular vesicle is just the carrier of information. So we are very interested in identify what are the molecules that can be carried by this extracellular vesicle. Our programs have been focused mostly in proteins and small non-coding RNA like microRNAs for the last 10 years. But now also we have been focused in DNA as well. So we have a very strong biomarker program running, and this is the stuff that we have been doing for the last several years, trying to identify molecules that can be carrier for this extracellular vesicle. In the context of cancer, identify extracellular vesicles that are released from the cancer cells and have a specific content, and that content is a reflection of the state of the disease. The other area that is particularly interesting for us as well, and it's a very hot topic, is the use of extracellular vesicles in therapeutics. This is something that we have been developing for the last 2 or 3 years. And essentially, you are interested in use extracellular vesicle for therapeutics. You have essentially 2 options, right? One is use the vesicle as a carrier of your therapeutic agent that can be for example, chemo drug can be a specific protein, a specific small molecule, etc. Or you can use actually the vesicle as your therapeutic agent. For example, there are plenty of information now on different clinical trials using mesenchymal stem cell derived extracellular vesicles for therapeutics. So you can see this is a kind of overview of our program and how we work in terms of the extracellular vesicle, right? So hopefully, I know that probably, first of all, I kind of talk a lot, so just keep the question for the end. And the other thing is, hopefully, you're gonna also trying to get this message at the end of the presentation. So what my lab does is trying to understand cell-to-cell communication through this extracellular vesicle. And obviously, when we talk about cell communication, we're talking about information that can be delivered from one entity to another. So it's very simple to explain with, for example, when you send a letter, you have the envelope, in this case, it's the extracellular vesicle, for example, and inside of this envelope, you have the letter, which is actually are the molecules that inside of this vesicle, and then you use it to communicate the message from, one side to another side. In this case, how cells can communicate with other cells. The other thing is quite interesting in extracellular vesicle research, and it's a big question in the field, is, for example, cells that are released, so vesicles that are released from cells, these vesicles are already predetermined to interact with a specific target, for example, or essentially the interaction is quite random. So it's something that we have been investing quite a lot of time the last few years, trying to understand how we call something that we call the barcode, which is here, of the extracellular vesicle. Because if you are able to identify, for example, vesicles that are more suitable to interact, for example, with immune cells, you might be able to encapsulate, vaccines inside of vesicles, right? or if you identify vesicles that potentially can be interacting more or have more affinity to interact with tumor cells, you can use also for therapeutics as well, right? I have a very simple video that I use all the time for just to establish some collaboration, especially in the hospital. So you can see that vesicle, essentially, the vesicle can carry information, and that information usually are bioactive molecules, including protein, lipids. 80% of the vesicle, they are lipids. So lipids is something important that have been overlooked through the years. And of course, nucleic acids, including, we focus a lot on microRNAs, but there are other types of no-coding RNA that can be also carried by this extracellular vesicle. And also DNA can be attached to the membrane of the EVs as well, which is quite interesting as well. So one cell can communicate with other cells through these extracellular vesicles, and this vesicle will interact with the cells, release the content, and modify different function on target cells. There are a lot of information about immune response, intercellular communications, metastasis, and angiogenesis as well. So there are information about that the molecules that are carried by this extracellular vesicle are bioactive and are able to interact with other cells as well, right? However, obviously, probably some of you have heard about extracellular vesicle, commonly known as exosomes. I'm going to discuss a little bit the difference between the terminology later in my presentation. And it's a lot of information about the effect of EVs in cell biology. So actually, we're quite confident now that extracellular vesicle communication and signaling is quite important in terms of cell biology, and especially in cancer biology. But unfortunately, even though we know that have an important role in cancer biology, that have not been translated in clinical applications, which is probably normal because even though extracellular vesicles have been described for many years ago, it's just in the last 20 years, and especially in the last probably 12 years or 10 years, it's have been this explosion of research on extracellular vesicle. So in general, it's still in early stages of development, right? And I think one of the problems that we face the people working in extracellular vesicles is about the diversity as well. That can be an advantage, of course, but also can be an issue when you're trying to identify, for example, specific biomarkers associated with extracellular vesicles, and also identify what are the molecules or the type of EVs that can drive your therapeutic effect on target cells, right? And this problem actually is not new. This is something that actually have happened in other fields as well. So I would like to just point it out and start with this. Probably you heard about the name of Ernst Starling, who actually was the first person that coined the term hormones, right? And actually this term is a generic term, or was a generic term for a specific chemical substance that control body fluids, right? And Sterling described the first hormone, that was the adrenaline, right? However, the full clinical benefit of adrenaline for management of surgical bleeding was not realized until the adrenaline was isolated, was characterized, and synthesized, right? So I feel that this is something that is common with the extracellular vesicle as well. One of the advantages, obviously, is the different function that different type of extracellular vesicle can have in cell biology. But on the other hand, it's important to identify the specific function of different type of extracellular vesicle. And for that, we need to improve, obviously, how we isolate them and how we characterize them as well. So you can see that this is not something that didn't happen before. And you have a good example, which actually with the first hormone, adrenaline, right? So I mentioned that diversity is a key characteristic on the extracellular vesicle, and if you check the International Society of Extracellular Vesicles, the definition for extracellular vesicle is a generic term for particles that are naturally released from cells that are limited lipid bilayer that cannot replicate, right? EVs are released from all living cells, are very diverse in terms of the size. So you have a small EVs like around 30 nanometers, and then you have bigger EVs like apoptotic bodies, which is over 1,000 nanometers in diameter. So you can see just the size is quite heterogeneous in terms of the release from the cells, right? Size probably is not one of the key characteristics of EVs. The biogenesis seems to be more important than actually the size of the extracellular vesicles. And you can, just to simplify this, you have essentially two main signaling pathways for biogenesis. You have vesicles that can form from the plasma membrane and then are essentially released to the extracellular environment. And you have vesicles that have a more specific type of biogenesis. That are formed inside of the multivesicular bodies as an intraluminal vesicle, and that intraluminal vesicle will be released to the extracellular environment, and that are the vesicles that we call exosomes, right? However, it's the size is the only characteristic, and I'm gonna point out this a little bit further in the presentation as well, right? And obviously, this extracellular vesicle can be released from cells and interacting with other cells and deliver the cargo, right? In terms of the applications of extracellular vesicle, I would like to point it out mostly four main applications. One is they are very attractive as biomarkers of disease because seem to be that extracellular vesicle is a fingerprint of the metabolic state of their releasing from the cell which are released this extracellular vesicle. So in terms of, for example, the cancer, if you're able to identify extracellular vesicles that are coming from cancer cells, you potentially the content of this extracellular vesicle will be a good representation of the metabolic state of the cancer cells, right? Which is very attractive. Also, they can be used for target for intervention. For example, this extracellular vesicle, this common characteristic that you observe in high metabolic cells, which are release more extracellular vesicle. If that happens in cancer cells, for example, you might be able to target that signaling for a potential therapeutic intervention as well. As I mentioned, they can be also carrier of information, and you can include some therapeutic agents in the extracellular vesicle for delivery as well. So there are several advantages over traditional soluble biomarker as well. And biomarker is something that we have a strong biomarker program focused in extracellular vesicle in my lab, right? And one of the key characteristics of that is that contain information about the cell of origin, as I mentioned, right? Also, the protection, because they're quite small. You're talking about that the majority of the vesicle in terms of the number are probably around between 100 to 150 nanometers. So they're quite small, and then they are limited by layers of lipids. So they're quite strong in terms of the protection for the molecules that are inside of this vesicle. And that makes them very attractive as a non-invasive or low-invasive potential biomarker, right? The other thing that is important, and we have been observing that in our studies as well, is because if you are able to identify molecules that are carrier for the extracellular vesicle, that also can increase the specificity and sensitivities of your biomarker panel as well, which is quite interesting. If you're interested in probably learn and know more about different type of extracellular vesicle, I suggest this particular review that we published a couple of years ago, in which we describe what is the state of the art in terms of the extracellular vesicle research, focus mostly in endocrinology, because this is something that we publish in behalf of the Endocrine Society. And you can see very useful cartoon, for example, this one. So here you can see that there are different types of extracellular vesicle. You can classify them according to the size, as I mentioned, as a small EVs versus large EVs. But probably size is not the main characteristic of these extracellular vesicles. You can see that there are vesicles that are formed here in the endocytic compartment by the early endosome, late endosome, and then the multivesicular bodies. And that are the vesicles that we're going to call exosome later on, which is around 100 nanometers in size. And one of the reasons that they are 100 nanometers in size is because the size of the EVs that are coming from this signaling pathway are limited by the size of the multivesicular body. So they are small EVs. But not all the small EVs are exosome. You have also small EVs that are ectosome, similar size to the exosome, but are released from the plasma membrane, right? So you're interested, I suggest please take a look at this particular review. Obviously, if you understand the characteristic of the extracellular vesicle, the biogenesis as well, you're going to be able to separate them and isolate them, right? And this is something that also is still continued discussion in the field about what is the best method, for example, to isolate different type of extracellular vesicle. But from my point of view, I think that also it's important to understand what other extracellular particles are actually present in biological fluids or in the cell condition media when you're trying to isolate extracellular vesicles. And this is something that we just published in Placenta, in which can give you a nice overview of the different extracellular particles that are present in biofluids, right? Not all the extracellular particles are obviously extracellular vesicles, so you can see that actually might be a small proportion of all the extracellular particles that are circulating there as well, right? And the majority of, might say all, the current isolation method to enrich population of extracellular vesicles is more likely that also you are co-isolating the other extracellular particles that necessarily are not extracellular vesicles. So you just need to be careful with what method you're gonna be using or choosing for your extracellular vesicle research, and have to be probably aligned with what is the question that you're trying to answer. For example, for our biomarker projects, what we're looking at is reproducibility. So hopefully, even though we can have some contamination of non-vesicle material, hopefully we are always isolating the same type of extracellular vesicle. If you're interested in therapeutics, probably what you need to check and what is interesting for you is batch-to-batch variation and the method that able to scale up the production of EVs, but because you will need a lot of EVs for therapeutics, right? So you're interested in determining which is probably the best method to isolate extracellular vesicle. Again, it's depending on what is the question that you're trying to answer. I really like this cartoon because here the author, what they do is classify the method according to concentration versus purification. So concentration means that quick, probably and dirty. So you get a lot of EVs, but you potentially we're gonna get a lot of non-EV material as well. Or method that you can actually get more pure amount of extracellular vesicle, but also you're gonna get less as well, right? So what I can tell you that because we have been working in the field for many years now, we try with all of them, right? And currently they're very popular. We are using a lot of cyclotron chromatography to isolate extracellular vesicles. But one of the issues with the cyclotron chromatography is very time consuming, and the translation of this particular method for routine pathology lab is not good, right? Probably you're thinking in translation and clinical application method that include a magnetic bead with immunocapture technology will be more suitable for clinical applications. Again, if you're interested in learn more about what is the best method that for your research in terms of the extracellular vesicle, you also can go back to this review and check according to the different method that we described for isolate extracellular vesicle or enrich for extracellular vesicles, and what are the conditions that are important for you. For example, if you are working therapeutic, scalability is something that potentially will be important for you, right? As I mentioned, also you can see that usually method that you can have a high amount of extracellular vesicles, so high recovery, are associated with a low specificity because there's a lot of non-vesicle material as well in the preparation. So it's something that you need to be careful as well, right? So just a summary for this part, for very brief introduction about extracellular vesicle. So extracellular vesicles are vesicle mediators released by cells, and they are released into biofluids, right? Also, they can carry a diverse set of different molecules, so they can involve in cell-to-cell communication, right? They also are released under normal conditions. So normal cell also release extracellular vesicles, secrete extracellular vesicle, but seems to be also that release can change and the content of the EVs can change in response to different microenvironment, right? They are very attractive for biomarker, for non or less invasive diagnostic, and also there are potential novel biomarkers associated with disease phenotype across a range of different metabolic disorder. And I'm going to focus mostly in what we do in our research in terms of the ovarian cancer research, right? Okay, so that was my brief introduction for extracellular vesicle, and hopefully now essentially I'm gonna go through the potential application of these extracellular vesicles. So I'm gonna be talking mostly or uniquely about ovarian cancer research and how we have been characterizing extracellular vesicles from different ovarian cancer cells, but also characterizing the circulating levels and content of extracellular vesicles across ovarian cancer progression. I put this image of a pregnant woman here because this is actually how I start my training. Actually, I'm a biochemist by training, and I did my MPhil in cancer. But then when I did my PhD, I worked with placental cells. And actually, I used to isolate placental cells from a human placenta, of course, at the moment of delivery. And then when I finished my PhD, I started to work with ovarian cancer, right? So, but metabolic, you just focus in the metabolic activity of this cell. Actually, placental cells are quite similar to cancer cells. So also a smooth transition for us. And this is the other half of our program that because of the audience, I'm not going to talk today, but we do a lot of still placental research in extracellular vesicle. Probably we are one of the most productive team in pregnancy and EV research in the world. So this is something that we're still doing a lot in my lab. So what I'm going to focus on is mostly about ovarian cancer. And the reason that we focus in ovarian cancer is because I think we all agree that we know more now that we knew actually 40 years ago. However, even though it's extensive time in research in ovarian cancer for the last 40 years, there are more technology, now that the technology that we have available 40 years ago, that unfortunately have not been translated in a better survival for women with ovarian cancer, right? And one of the main reasons, or probably the main reason for that, is because we are not able to identify women early enough in the development of the disease. I really like this graph because it's very simple. So you can see here the 5-year survival rate for women with ovarian cancer and the different stage of ovarian cancer. So it's very clear that if we are able to identify women's early stages, especially stage 1, we're going to be able to actually save the majority of these women, right? Unfortunately, the majority of these women, 75%, are identified in late stage of ovarian cancer, stage 3 and 4, when you can see the 5-year survival rate is quite poor, right? So the question is how we can actually identify these women as early as possible. And we have been focused in the idea that potentially if we're able to identify changes in the extracellular vesicle signature very early in this oncogenic transformation of cancer, normal cell in cancer 1, we're going to be able to develop a test for early detection of ovarian cancer, right? So that is the idea, and that is the hypothesis. So what I'm going to do in the next, hopefully 20 minutes, 15 minutes, is going through different studies that we have published for the last 10 years, trying to give you some evidence about the role of extracellular vesicle in ovarian cancer. So we know that ovarian cancer cells can release extracellular vesicle, and the release of the extracellular vesicle and the content is depend of the invasive capacity of these ovarian cancer cells, and we have been demonstrating that in vitro, but also in vivo. And finally, for the last hopefully 7 to 8 minutes of the presentation, I'm gonna describe our study in characterizing extracellular vesicle in blood in terms of the ovarian cancer early detection space, right? So we have been able to develop a test for early detection of ovarian cancer, and this test have been actually been in the progress for the last 8 of 9 years. Our OCRF7 test, right, for early identification of ovarian cancer. This is just a timeline to just demonstrate how difficult have been this journey, and for a couple of reasons, right? And we are all in research, right? So one of the reasons that have been difficult is you have to be able to generate a lot of data to try to convince our different organizations that provide funding for our research. So we actually make a lot of progress early in our program with mostly philanthropic organization, and then when we have consistent data, we were able to actually convince our NHMRC, which is the main organization who give money here in Australia, to fund this particular project. And I don't think I have time to show you all this, but the only thing I'm going to mention is this is our study design. So we have been actually, this was probably around 3 or 4 years ago. So at the moment, we have over 1,500 patients involved in this project. And what we have been able to essentially to do was trying to develop a test and then validate this test in a total and independent cohort of patients, right? So I'm gonna jump this because I know that I don't have time. So I'm gonna jump just to the final bit, which is, so I'm gonna stop here a little bit. So this is essentially our discovery and training cohort. So you can see that we're able to identify, to develop a test with 98% specificity, mostly to identify women with early stage of ovarian cancer versus our healthy control, was essentially women without ovarian cancer. And this is the performance of the training versus the validation. So you can see it's quite similar, suggesting that our model is not overfitting the data, right? So essentially, this is the one. So you can see that this is the sensitivity and 98% specificity for our training and validation cohort, right? So you can see that a 98% specificity, which is the capacity to identify women without the disease, we're able to identify women with ovarian cancer with a sensitivity between 61 to 86% in range, with an average of 76%, right? Which is actually very good. So all this initial number of patients was for the discovery and training of the test. And then what we did was validate this test in a complete independent cohort of patients in almost 500. So the test was designed essentially for early stages identification of women with early stages of ovarian cancer. And what we have for ovarian cancer in general is CA-125 is using, you know, around the world for detection of ovarian cancer, which is actually very good for late stage of ovarian cancer, but it's not that great in early stages. So the first thing that we did was trying to determine actually what is the capacity of CA-125 to identify women with early stage of ovarian cancer in our cohort. And you can see that the classification efficiency was around 62%, right? So it's a little bit better than flip a coin, which is very consistent what is in the literature. With CA-125 actually is not very reliable for early stage of ovarian cancer. So then we apply our test to this cohort, and you can see that the classification efficiency was 98%, which was fantastic. So with this data, we went to the NHMRC, and now what we're trying to do is validate this signature in collaboration with Professor Usha Menon and Dr. Alex Gentry Maharat from the UCL to check if the signature can identify the cases of women with ovarian cancer in sample coming from the UK CDOC trial. So we're going to answer two main questions with this particular project. We're going to try to determine if our test actually can identify the women with ovarian cancer in this cohort, but also how early, because as you all know, this trial have sample from women at the moment of the diagnosis, but also up to 5 years before the diagnosis with ovarian cancer. At least we're going to be using up to 5 years before the diagnosis, which is fantastic for us, right? So as a message, hopefully I have been providing some evidence to convince you that extracellular vesicles have an important role in cell biology. Hopefully we're going to be using that information for clinical application in the future. And that can be used for early detection, for identify therapeutic targets, and also to identify how women can be responding to chemotherapy as well, right? I think one of the challenges that still remain in our research is how we can actually refine the EV isolation and cell specificity, especially for the and cell-specific targeting. I think that is the key for developing therapeutics, but also for biomarker. And the other thing is important is one of your goals is trying to implement whatever you are studying, make it available for clinical implementation. It's super important that for the very early, your research need to align with the different regulatory requirements, depending on in which country you are working with. I think that is super important when you're trying to commercialize a test in general, right? I'm almost done, so just a big thanks to my group. You can see that this is a lot of people involved in this particular program. This is our photo for 2024. I would like to just mention one of the key personnel here, Dr. Andrew Lai, have been working with me for probably 7 to 8 years, and he's the team leader for the proteomic area of our program. And so he's dealing with all the proteomic analysis in the extracellular vesicle. Then Dr. Dominic Wanson is looking after the genomic program in my lab, and he is responsible for all involved genomics EVs as well, right? Also, we have more staff. Samya is looking after all the therapeutic part of the lab as well. We have more younger researchers coming from all over the world to work with us. So I'm very grateful for all the people working with me, of course. And other thing that is super important for our program and for everyone is the funding. So we have been able to progress in our research program thanks to the funding. Mostly in our initial stage was from philanthropic organizations, including the Lion Maker Research Foundation and the Ovarian Cancer Research Foundation. And in the last few years, also we received significant funding from the NHMRC and the MRFF as well, which is fantastic. Just to finalize, I'm going to mention that the university have approved the establishment of a new center, which is the UQ Center for Extracellular Vesicle Nanomedicine, which is an effort for over 30 different research groups in the university. And over 70 researchers are involved in this initiative. So extracellular vesicle is something that we're going to be very focused on as university in the future as well. And hopefully our website is going to be done in the next few weeks. And then hopefully we're going to be able to establish also more collaboration, not only across the university, but overseas as well. So if you are interested in extracellular vesicle research, send me an email and I can put you in contact with the different groups across the university as well. Thank you. I know that was a lot of information, but hopefully you enjoyed that. I'm more than happy to answer any question if I can. Thank you.
Great. Thank you so much, Carlos. A really elegant presentation on EVs. So I'm going to open the floor for questions. If anybody had any questions or comments.
Any question is welcome.
Have ideas for collaborations?
Yeah, well, I can probably start with just mention that we are collaborating with Alex and her team at the UCL in this ovarian cancer early detection program. But we are open to new collaboration as well. So if someone have a cohort of patients that they want to provide some extracellular vesicles, we are very open to collaborate as well.
Great.
Alex, it's
I
almost
know we are
oh, sorry,
we
Alex,
are
it's
almost
almost 10. Yeah, but yeah, please
we
go
are
ahead.
almost there. Carlos, can I just ask you a very, very brief question? So if you
Sure.
think about different cancers and the extracellular profile, how similar or different would they be? So will the learning from ovarian cancer be applicable to breast cancer and pancreatic cancer, or will you really need to work in sort of silos and work packages across each of the cancers?
Yeah, that's a very great question. I'm probably the honest answer I don't know, right? What I guess is every cancer will have a different signature in the extracellular vesicles. That is something that we think that is happening because the proteins and the microRNAs that we have been working on, some of them are quite specific for ovarian cancer, mean that they haven't been associated with ovarian cancer before. So, and you can see similar patterns in extracellular vesicle research in other type of cancer as well. One of the problems that we face as well in our research is the majority of the time what you do in vitro, what you're trying to do in the animal models, it's very hard to translate it into patients, right? So it feels like that if you really want to develop a test for biomarker, probably obviously your in vitro and your in vivo data will provide some rationale and evidence that actually these EVs are doing something, but you might need to do a complete EV analysis in patients, and that analysis might be specific for different type of cancer as well. And that is something that we are very interested in. What I can tell you that we have a project under review that we are planning hopefully to profile our EV tests in different cancers as well to determine if this test is specific for ovarian cancer or potentially can be applied to other cancers as well, which is, I think, is important. It's a very important step to determine the intent of use of our biomarker panel for commercialization.
Great. Thank you so much. Don't think there are any questions or comments. I'll hand over to Latifa for the last word about the next webinar, but I would like to thank everybody for attending today's seminar and for Carlos presenting this at midnight, Brisbane time.
Actually, it's 1 a.m.
Now close to, yeah.
So yeah, okay. Thank you, Alex, for introducing the speaker and for moderating this session. Thank you, Carlos, so much for accepting to present so late. I'm gonna say night, but it's early morning. Actually, it's now it's Friday over there versus here we're still in Thursday. But anyway, I put on the chat the upcoming webinars. It's about All of Us research program that is going to be presented by Dr. Jeffrey Ginsberg, which is scheduled on March 28th at 9 a.m. EST. I know sometimes the time is not convenient to everyone, but please make sure that we send to watch the current and previous webinars in our websites, and I will be sharing the recording to others who missed this exciting and informative presentation. I personally learned a lot, especially a friend of mine just lost his wife from ovarian cancer 2 days ago. So please, when we send you the webinar, share it with your colleague and others so they can join and learn from those exciting presentation. And again, mark your calendar for this exciting 1 million across the US All of Us program, which is inviting 1 million people across the US to help building the most diverse health database in history. So again, stay tuned for the announcements and looking forward to seeing you on March 28th. Thank you again. And have a great day.
Thanks. Bye, everyone.
Bye-bye.
Thanks, Carol.
Bye, everyone. Thank you.
Thank you.